Hello students, I think it is better to have this virtual classroom again with a better quality of the sound and also the picture. So today I want to talk about the nucleic acids, and so the nucleic acids are another group of biological molecules. Nucleic acids or another type of the biological molecules and are made of the nucleotides. So nucleic acids are made of the nucleotides, okay, and also there are three molecules that are made of the nucleotides that we want to talk about them. Three molecules that are made of the nucleotides. One of them is DNA, the other one is RNA, and the other one is ATP. This one is called as deoxyribonucleic acid, and this one is ribonucleic acid. This one is adenosine triphosphate. Or called as ATP. So we know that we start first with the DNA structure, before that, because all these molecules are made of the nucleotides, I want to first talk about the structure of the one nucleotide and to see how actually it looks like. So one nucleotide this is the structure in a very simple way. So I first draw a circle to show the phosphate group. So this is phosphate group. Now this phosphate group, this is connected to a sugar molecule. The pentose sugar. This pentose sugar from one side is connected to a base. Okay? So DNA structure may look very complicated, but actually it is very simple. If you just break it down, you will find out that it is actually made of some simple components. And it's the shape of this DNA molecule finally will be a double helix, as you know. It's like a ladder and is a strand of a DNA and also a polymer which is made up of repeating units of the nucleotides that you see here. So these nucleotides will be repeated and repeated and repeated to make one DNA molecule. Nucleotides have 4 different various and versions, and when are actually joined in a very long chain, they make a strand. So this is one DNA. So the DNA molecule is a double helix. This is one strand, this is second strand, and these strands between them, there are bases between them that joins them together. Okay, so for example, this is tripod, and here again 3' goes to 5'. So this is the structure of the DNA molecule. I just want to first show you how one nucleotide looks like. So one nucleotide joins to the other and the other and the other and the other. It makes one strand of the DNA this way only. So this is, I'm just showing to you how one strand of the DNA is actually made. Now we know that the nucleotides are having three parts, and one of them is the phosphate group. So we show the phosphate group by that circle. You do not need to actually to show the whole structure and the chemical bonds and everything, but I just want to show you how the phosphate group actually looks like. So is 1P, one phosphate, attached to 4 oxygen. This is double bond, and here this is the oxygen. These two oxygen, the charges are a little bit actually minus, and so from here, from this oxygen, this is where the phosphate group attaches itself to the sugar molecule. I said that the sugar molecule in the DNA is a pentose, and also in the RNA also is a pentose sugar. But what is the difference between the RNA sugar and the DNA sugar? Okay, in the DNA sugar first. This is for DNA sugar. So I want to show you how actually it will look like. Carbon again, and again, carbon, carbon. So okay, here we have CH2OH, but even the reaction actually CH2OH we have here, but it's gone already, is bonded together. So then what is happening here is that what is important here are the OH group, hydroxide, and also the hydrogen group here, and hydrogen here down, hydrogen up, hydrogen OH, and here is H. Actually it was CH2OH before. Okay, so what is happening? This is a deoxyribose in the DNA. This is deoxyribose in DNA. But in the RNA, what is happening? The sugar is exactly the same, would be the same. Okay, and from here we have, okay, CH2OH, and here we have H, we have H, OH, we have OH, H. Okay, now here is very important. This is H, and here is OH. This hydrogen and OH group here, and the second carbon, these are very important. In RNA, the H is replaced by the OH. So we have ribose sugar. So these are all the differences between the ribose sugar and the deoxyribose in the RNA and DNA. So both of them, they have the same structure till now here, up to here, but the only difference in their sugar is that in the DNA we have the deoxyribose, this one, which is H here, and in the RNA, this one is replaced by OH. So this is ribose sugar. This is called as sugar phosphate backbone. Now, what actually now we are looking for is the base actually which is attached to this part of the sugar. Let's have a look into the different bases that we have in the DNA and also maybe in the RNA. Thank you. In DNA, we have 4 possible different bases, okay, that come into 2 different categories. So we have 2 different types or categories of bases. So first I say in DNA, because the structure of the DNA and RNA somehow are the same. As we say, they are made of the nucleotides, but what actually is different, as I mentioned first, is in the sugars, the pentose sugar. As you saw, the structure of the pentose sugar in the DNA, on the second carbon, it has hydrogen, but in the RNA, it has OH. So let's see another difference between the RNA and DNA structure. So look at the bases now, the bases that are attached to that phosphate sugar backbone. Okay, we have 2 different categories of the bases. The first one is called as pyrimidines. And the second one is called purines. Okay, so what are the differences between these 2? The purine bases structure is 1 ring. Sorry, pyrimidines are 1 ring. And the purines are 2 rings in their structures. Okay, now let's find out what bases actually we have that goes under pyrimidines and which bases they go under purines. The name of the bases that we have that are made of 1 ring only, they are cytosine. Remember, because if you want to remember, and don't forget, because that's a pyrimidine, so it has a Y, so cytosine also has a Y. So because both of them have Y, so they are under the same category. So you won't forget this one then. So the next one is, which one? This is, the next one is thymine. Also this one, thymine. Thymine also has Y. So these 2, because they have Y, so they go under this pyrimidine that has Y. So both of them are 1 ring actually bases. Now we go to the purines. Purines, they have 2 rings. Let's show which bases they go under this category. The first one is adenine. Adenine. You can show later by a symbol. For example, for cytosine, you can put C. For thymine, you can put T. For adenine, you can put A, capital A. Now, for the other one, which one is it? It is guanine. You see, guanine and adenine, they don't have Y in it. So you can easily know that they do not belong to pyrimidines. So they belong to the purines, and they have 2 rings. Now, what is the difference between the structure of the DNA And RNA is this. It rests in this, that in DNA we have cytosine, thymine, adenine, and guanine, but in the RNA structure, the bases. Okay, we have cytosine. Do we have thymine? No, we don't have thymine. So let's continue. Do we have adenine? Yes, we have adenine. How about guanine? Yes, we have guanine. So what is actually added here is another base, which is called as uracil. We'll show why you. So the one which is added here is uracil. You can find it in the RNA nucleotides, okay, in the bases. So we don't have thymine in the RNA. Another difference between RNA and DNA structure. So you have to remember. Now, let's continue and see the structure. You do not need to know the structure in detail of each basis, but I just want to show you. I want to first start with the structure of the cytosine and thymine because there are pyrimidines that have only one ring in their structure. So let's do that together. So I bring the paper a little bit closer to myself. So the first thing in this, I start with the cytosine. Okay. This is N. This part also is N. Wherever I don't put anything, it means that here there are C or carbon. Okay. Here is a double bond connected to oxygen, and here is a double bond again. Here we have double bond again, and here we have also NH2. This is the structure of the cytosine, and from here, this base is connected to the sugar, so this is cytosine. Okay, now let's go for the next one, the structure of the base called as thymine. 1, 2, 3, 4, 5, and 6 corners. Then here is the double bond. Nowhere else we have double bond. Here is NH. Here is nitrogen alone. And here we have one CH3. And here we have a double bond connected to the oxygen. And here we have a double bond again connected to the oxygen. And from here, okay, again, the base should be connected to the sugar, to the phosphate sugar backbone. So the name of this one is thymine. So both of them, these two, they are pyrimidines. But because they have one ring only in their structure. Okay, so now we have done with these two. Now I want to draw the structure of the adenine. Adenine has two rings. Okay, so 1, 2, 3, 4, 5, 6. The first ring has 6 corners here and here. So here is nitrogen. Here is nitrogen. This is NH2. Okay. Now, from here, it is connected to another ring of it. So, there we go. This is nitrogen. This is double bond. A double bond also we have here. Okay. So, we have another one here, nitrogen. So, now, here it is connected to the phosphate sugar backbone, and it's called as adenine. Now let's go and do it for another base. These bases here that I am doing for them is the purines. The purines, they are made of two rings. Okay, so again, for the next one, or the last one, guanine, double bond, oxygen, NH. Again and again, here's double bond. Again, we have nitrogen. Again, double bond. Here we have NH2. Okay, so again from here, this side, we have nitrogen. We have a double bond here, and then we have nitrogen, and then is over. This is 5 ring. This is 5 corners. This is 6. And then from here, which is the nitrogen actually that are available, this is connected to the phosphorus sugar backbone. So this is guanine. Okay. So these are the structures that you may know. I just did for the uracil too. So it's a 1 ring base, so it belongs to the pyrimidines category. Now look at these 2 nucleotides, one here and one here. They are connected from this part together. But how they are connected? You see, each nucleotide is linked to the next nucleotide by a condensation reaction. If I can just write here for you, condensation reaction. So this is very important. This type of the reaction is a condensation reaction. Why is this condensation reaction? Because during the reaction, a covalent bond is made, and also it removes water. So it removes water from this bond. So one water molecule is going out here. H2O is formed here, okay? So that's why we call it as a condensation reaction. And also the bond which is formed here is the name of the bond. What type of the bond is formed is phosphodiester. Phosphodiester bond. So phosphodiester bond, but what type of the reactions? Condensation reaction. You can see here we have cytosine, we have guanine, and which is connected to the phosphate sugar backbone. These phosphate sugar backbone are connected together. Here you see, from this is their first carbon, second and third. From the third carbon, the oxygen of the phosphate group is actually reacted with this hydrogen, and then it produces a water molecule. So during the condensation reaction here, and the bond which is formed here is a phosphodiester bond. And this actually continues, just continues until this is one strand of the DNA, okay? There's one strand only. This is one strand of DNA. But you know that the DNA is a double strand, helix, in a helix form. Okay, so we all just have made one strand structure. So still we need to draw another strand to show the real structure of the DNA. As I told you, you do not need to remember and memorize all those details of the molecules, how to draw. So what you are supposed to do, you can just simply draw them. Let's see how you can draw one strand of this molecule. Okay, I show, for example, by this circle and blue color, this is the phosphate group, okay? And from here, it is connected to the sugar molecule. I put this one because this is oxygen here. And the rest are carbon. You can just remove it. If you do not put it, even is okay. And then we have a base here. For example, our base is adenine. Then from here, again, we have to make a connection from here we have to make a connection. We have phosphodiester bond, and during the condensation also the water is released, so we have another phosphate group, and this phosphate group is connected to another molecule of the sugar, as you can see here. So again, from here also the sugar molecule is attached to another base, for example guanine. So the next one, again, this sugar also discontinues the scenario, as you can see, again attached to one phosphate group. Phosphate group is attached to one sugar molecule. This is one strand, one strand of the DNA molecule. So it is attached from here, for example, to another base, for example, adenine again. So I just draw another one. Let's do not mismatch. Okay, I'll continue with the same color. So we have again phosphate group. Phosphate group is attached to this sugar, pentose sugar, and the sugar also is attached to a base, which is, for example, thymine, and it just continues again. You can draw more. Again, phosphate group attached to a sugar molecule, and then attached to, for example, a base called as cytosine, and more. So this is one strand of this molecule. The polynucleotide or nucleic acid, one strand of the DNA it is, okay? So DNA, as I said, is a double strand molecule. So we need an opposing strand just in a reverse direction, in an opposite direction or anti-parallel to this strand. So what we have to do, we have to draw another strand, another chain of the nucleotide just in front of this, but in the opposite direction. So again, let's start. I have a base here. This base is connected to the sugar, one sugar molecule. You see that? The sugar also is connected to one, for example, base. Now, this is very important what to put here. I will first draw this strand, and later I will fill inside the base box, okay? I will tell you how to fill it. So the next one comes from here again. From this side, we have a base attached to this sugar molecule, and from here it is attached to another base. I will write the name later. Again, we have another phosphate group attached to a sugar molecule, and then this sugar molecule also is attached to a base. Again, this continues. We go on and on and on. This is the one, and here we have one sugar molecule again, and here is the base. And again, until we finish. Yes, another pentose sugar. There we go. And that's it. So that's what you can see. We have another strand anti-parallel to the first strand. Okay? Now, what should I write here? Which bases I have to write here? Anything can be written here? I don't think so. The bases, you should know that, always have to be paired up. Okay? So a purine base always pairs with a pyrimidine base. A purine base always with a pyrimidine base. It means that C always goes with the G and T always goes with A. This is very important. You cannot just put everything there. So it means that the A always pairs with the T and C always pairs with the G. Okay? So let's see now, based on this definition, fill the boxes here. So I said that the C always go with the G. Sorry, I have to write in the opposite direction. It's very hard for me because the strand is anti-parallel. So here is G. If it is T, this one should be A. If this is A, this one should be T. If this is G, this is C, and A should be T. Now, let's see. This is how the double helix is now formed. Okay, this is very important now. We need to explain the direction of the strand. Okay, we should know, or we have something like to explain in a way that which direction each strand is. In order to do that, we use the term 5' and 3'. 5' and 3'. These are the terms that we use. I think it's better to use a darker pen. So we use 5' and 3' to show the direction of each strand. So where should I put 5' and where should I put 3'? The end, which ends with the spur phosphate sticking out, is the 5'. Okay, so this is the one that has the spur phosphate group, which is sticking out of the molecule. So I just name it as 5'. So of course the other end that doesn't have it, I just name it as 3'. And also for this side, this one is hanging out. So I just name it as 5'. And the other end that doesn't have it, I name it as 3'. Now, till now, our actual shape is getting more completed. Another important thing is that there are hydrogen bonds connecting these two strands together, so the ladder won't actually be just detached easily. Something should hold these things together. These are hydrogen bonds between the bases, okay? But how they are the hydrogen bonds that join the two strands together between the base pairs, between A and T that we have here. Okay, you can see we have two hydrogen bonds. I showed it like this. So there are two hydrogen bonds between A and T, adenine and thymine. Okay, there are two hydrogen bonds. Between these adenine and thymine also we have two hydrogen bonds. Here also we have two hydrogen bonds. Okay, now how about these two? Are they the same? No. Between these two, which is guanine and cytosine, we have three hydrogen bonds. Three hydrogen bonds. 1, 2, and 3. And also here, 1, 2, 3 hydrogen bonds. This is very important and you need to remember. Let's have a look into this diagram and see what actually components we can see here. So this is the phosphate group. This is one nucleotide. This is second nucleotide, and this is one strand, one DNA strand. This is the second DNA strand. Okay. So, for example, if this is 5', this should be the 3'. end of that strand. So it's like this, is continuous. Okay, so the first thing that you have to pay attention is the phosphodiester bond here, which has formed between each of the nucleotides. So this is the bond, okay, the connecting between these two. Also should be one phosphodiester bond. So here it is, okay, this is one phosphodiester bond that is during the condensation reaction, water molecule is produced, and these are the bases. This is the phosphate group, and here, for example, we have thymine, and we have here guanine. Okay, so and another thing that you have to pay attention is that this one, okay, these are two nucleotides from another strand, and this phosphate group is connected to the sugar group by this phosphodiester bond again. This is cytosine, and this is adenine. Now, what are they? There are those hydrogen bonds that are formed between each of the bases to connect the two strands together, two strands of the DNA. Look at this. This is thymine molecule, and this one is adenine base. Okay, so I said between the adenine, always T goes with the A, so this is a correct actually combination, George. And also the hydrogen bond between the thymine and adenine is only 2. So, but here we have guanine, right here guanine, and here we have again cytosine. Cytosine always goes with the guanine. So one ring goes with the 2 ring. One ring goes with the 2 ring. Don't forget this. So the bond, hydrogen bond between guanine and cytosine is 3 hydrogen bonds, as you can see here. Okay, so these are very important. This is the base this is the sugar phosphate backbone, phosphate sugar backbone. This is the base. This is one strand, and also we have sugar phosphate backbone, and we have base and hydrogen bonds. These are the base and hydrogen bonds, these parts, and these are sugar, pentose sugar, and phosphate. So we call it as sugar phosphate or phosphate sugar backbone. Don't forget this one too. So now look at the structure of the RNA. What are the differences between the RNA and DNA structure? These are very important for you to know. The first thing is that the DNA was a double actually stranded molecule. It was a double helix, if you remember. So it was like a ladder with the 2 parts, and the 2 parts are just twisted, wired together. So but here is only one strand, okay, but so single stranded molecule. And the second difference is that in this RNA, instead of the deoxyribose, we have ribose sugar. So look at the differences I told you. Everything is the same, but in the ribose sugar, here is OH, but in the deoxyribose, this one is hydrogen. So that's the only difference. And when they attach from here to the phosphate group, from here the OH reacts with the phosphate group, so it goes away, so becomes CH2. And when it reacts from the other side also with the base, so OH group also from this side will disappear. So it reacts with the base group. Doesn't matter which base it is, but the OH reacts with that base. It reacts with the base, and this one reacts with the phosphate. Okay, and another difference between the RNA and DNA is that the DNA had thymine base, but in the RNA there is no thymine, and instead it has uracil. You remember that the uracil also is a pyrimidine, is a one ring base that I, the structure I already have shown to you. As we said, we have different RNAs. We have mRNA, which is messenger RNA. We have rRNA, which is ribosomal RNA, and tRNA, which is transfer RNA. Now, we want to go back and have a look into semi-conservative replication of the DNA first during the S phase and see how the DNA is replicated. We just want to go back to the DNA structure. So in the cell replication, we don't want to put half of the DNA in one cell and half of the information into the another cell. So it means that so otherwise, if it continues, just we will go. Finally, at the end, we won't have any information or the genes left in one cell to be replicated. So DNA first needs to be organized so it is easier to move around for cell division. So it forms a structure called chromosome. And also, of course, the molecules, they wind around the histones, they form nucleosomes and the supercoils, and then become denser, they form chromosome structures. At first, the chromosomes are single, but they become double later, and they are joined from the middle, if you remember from the last session. So they attach to each other from the centromeres. To copy the chromosome like the DNA, so the DNA needs to actually go into a process which is called DNA replication. This is our topic now. So you need to remember everything about the DNA structure. Better to go and recap it and review whatever I have told you. Now we go to the stages of the DNA replication to see what happens now. I said that the DNA is a double, actually stranded like this, a helix. And there are nucleotides here. They are joined together by the phosphodiester actually bond, and here we have bases. The bases are joined together by hydrogen bond. Okay, the hydrogen bond is connecting these two strands together. Now, during the replication, in these two strands, they first need to unwind and unzip. Okay, so this is done by an enzyme which is called DNA helicase. DNA helicase comes and opens up, it moves like this, and opens up the strands like this. So what does it do actually? It breaks the hydrogen bonds between the bases and causes the molecule to be unzipped and become separated. And so by this actually action, the DNA helicase produces a template and make the DNA ready for replication. There are templates that now, so DNA helicase is just actually opening these helix up by breaking the hydrogen bonds. So there are the templates, the parental. So this is original template, and also this one. Okay, so just unzip it and make it ready for the replication. You have to notice, you have to know that always the replication happens. We say that by, okay, DNA helicase opens it up, but the replication always is is actually the replicated DNA is made from 5' to 3'. Okay, so it means that that end of the DNA, the template, the original template, which carries 3' and it is free, so that's where the actually replication starts from. But the other end, so cannot happen here. It cannot happen here. We cannot go at this direction because always these replication are made from 5' to 3'. So this is not possible. Then we have to go to the opposite direction from the left to the right. So how is it possible if I go from the left to the right? So how about the rest of the molecule that is not been replicated? So that's what I want to talk about. As you can see, this is the leading strand, okay, because normally they when the DNA helices opens these forks, is shaping, and we have these templates. This is the original template, and we have this leading template. Why leading? Because that's where actually that replication happens. So the DNA polymerase 3 now, DNA polymerase 3 actually will move along from 3' going to 5' and follows the DNA helicase exactly, and are going along the leading strand template and synthesizes new strand from 5' to 3', as you can see. It's very easy now for it, and goes okay, and add up, just follows the DNA helicase. Now when the leading strand is being replicated, the lagging strand template of the DNA is more complicated. This is the lagging strand, this one that has 5', and as it is very hard because the copy is made from always from 5' to 3'. So that template should be 3', but this one is 5', so it should be in the opposite direction. So some of the genes here will be left over. So what should we do? So this is very difficult actually to translate, to replicate. So this is a lagging strand. This is called as a lagging strand. If you want to lay in a 5' to 3' direction, it would be going in the opposite direction to helicase, okay, and also in the replication fork. So that's why it's very hard. What actually happens here, we have to do is that the replication happens in a very small little sections. So one section here will be replicated, one section here, one section here, one section here. This doesn't happen, all like, exactly in a direction of the all along the strand, the whole strand with the template. So how it is actually happening, an enzyme which is called RNA primase follows helicase, DNA helicase, and is actually unzips the double helix. The RNA primase leave a small section of the RNA called RNA primers, which show where the DNA polymerase should start its work. So how actually these things are happening? I said it's these replication are happening is very small section. So it is actually the job of the RNA primase. The RNA primase, okay, is making very small sections on the lagging template or lagging strand, which are called as primers, RNA primers. It means that it shows to the DNA polymerase where actually it should start replication. Okay, it shows it to the DNA polymerase. After that, DNA polymerase comes and attaches to the RNA primers. the one that is made by RNA polymerase, the DNA polymerase attaches itself to it and starts replication in the direction that it actually works, from 5' to 3'. Okay, and once the job is done, it detaches itself from the this template. Okay, so the job is done and it leaves and goes. Now we have a new strand of the DNA, but they are in fragments, small fragments of the DNA formed, which is made by the DNA polymerase. They are called as Okazaki fragments. These are small parts that are made by the DNA polymerase tree. So, but these Okazaki fragments are not alone, okay, on the lagging strand. They are actually accompanied with the RNA primase. So, actually these are extra. There are no need anymore. We have to get rid of it. So then the next job is to remove them, to remove these RNA primase. And what molecule is doing that? DNA polymerase 1. DNA polymerase 1. So what does this do? It removes the RNA primase, which is also attached to Okazaki fragments. Okay? So it removes them. It removes the RNA primers and replaces all the primers. So, but just what is left there is Okazaki fragments. Now we have only Okazaki fragments after the DNA polymerase 1 comes and removes all RNA primase. Now, the last job is to go along and just stitch those small bits of the DNA together. I mean these Okazaki fragments now, because they're alone now, they should be connected together. Now this is the job of DNA. This is the job of DNA ligase. What does DNA ligase do? It comes and connects all those Okazaki fragments that are made by the DNA polymerase tree together. So it uses ATP or energy to do this job. So DNA ligase uses ATP to do this job. So we have these two scientists, they have come up with the idea of the semi-conservative replication. Semi-conservative replication of the DNA. So, and also this scientist's name was Meselson and Stoll. These two. In 1958, quite a long time ago, they used coli, is a bacteria, Shirishella coli, and two nitrogen isotopes named as nitrogen 15 and also nitrogen 14. Okay, to demonstrate how the density of the DNA changes over the generation as the N15 isotope was replaced with the N14 isotope. Sometimes DNA replication makes a mistake about 1 in every 1 billion. 1 in every 10 billion nucleotides. There is a mistake always. So however, the DNA polymerase comes to proofread the DNA. okay, to solve this problem, to repair its mistakes that it has done to go along. Even when a mistake happens, another process comes, which is called as a mismatch repair. Again, after this, again, we have another process which is called as a mismatch repair. Can fix that problem. So it is an inbuilt mechanism in the cells which stop mistake. But still, mistakes will happen, errors will happen. So that's why we have mutations. There are mutations. And the last thing that I want to say about this part, just about the semi-conservative replication, is that I want to repeat again. So because this is a very long process, if it wants to start from one end and continue until it reaches to the another end of the DNA. So this replication happens in a various parts of the DNA. So the bubbles are forming there in the DNA. We call them as bubbles of the replication, or like this, as you can see. That's the DNA, and from here the DNA is opened, perhaps by the DNA helicase, as you can see. So it doesn't start from one end and go to the other end. It takes a long time. So in order to make it faster, that's what is happening. This is 3' end, 5' end, 5' end, 3' end. So what is happening is here we have replication forks and also replication bubbles. So finally, when the replication at each part is been done, they join up, the whole chromosome is replicated. So they join up together so that the whole chromosome is replicated. The two strands will wind up to form double helix shape. And this is called semi-conservative replication. We start with a molecule of DNA, and one of the original strands can be now found in the DNA of each of the two new molecules of DNA. So half of the original DNA is conserved into each of the new molecules. If you look at here, this is the DNA that we have started. This is the parental DNA, okay? And if I want to show with another color, so once it is replicated, it goes into two different cells. So this is parental, of course it's come from here, the parental DNA, and now we have the replicated one. So each of these cells, they receive identical DNA, which is exactly similar to the parents. So it comes from the parents. So so half of the original DNA is conserved into each of the new molecules, as you can see. So that's why we call this as a conservative, semi-conservative replication. Thank you very much for paying attention to me. And maybe I just make a very fast review on the whole chapter, those important parts, of course, of the replication. So the DNA helicase. DNA helicase was responsible for unzipping the DNA helix by breaking the hydrogen bonds between the bases. And so it opens it up like that. So with shaping those forks like this, making it ready for the replication. Then we have DNA polymerase 3. And this one forms phosphodiester bonds to form sugar and also phosphate backbone and join the nucleotides together, and also makes new strand from original strand template from 5' to 3', from 5' to 3'. And we have RNA primase. This RNA primase actually comes into the template, which is the lagging template, and works on it. Shows where the DNA polymerase should actually start on the lagging strand template. So it actually makes RNA primers and makes that part, shows that where on this lagging strand should be replicated. Show it to the DNA polymerase. So and also after this, when the DNA polymerase come and translate that one or replicate those ones, so we have a small fragment of Okazaki fragments, and plus RNA primers left on the lagging strand. After that, the DNA polymerase 1 the DNA polymerase 1 comes to remove the RNA problem. So it removes RNA primers. So what is left now? What is left is the Okazaki fragments. So after that, we have DNA ligase. And this is the last one that comes and joins those Okazaki fragments together to complete the that replicated DNA and the strand. And that's the whole thing, the whole story. Thank you very much. Hope you have understood it well. Please be in contact with me and ask me any question if you don't know any part of it. Thank you very much. Stay safe and healthy. See you again.